Hello, my name is Francisca Sanchez. Today is August 9th, 2018. It is week 6 of lab 6 of Bio 225. Today I'm going to be doing the salt tolerance and pH testing. So I've put on all my stuff. I've already disinfected my work area. And I also have labeled my nutrient broths. So I am going to go down and start with the cutting off the HCI and the NaOH. So I will be cutting these. Cut those in a 45-degree angle. And I'm going to light my candle so I can sterilize my test tubes. I'm gonna remove the S-Epi pH 5, remove the lid of that, and pass the tube, past the opening over the candle flame to sterilize it. Now I'm going to add exactly 1 drop of HCI to adjust the pH. So 1 drop, sorry, 1 drop. Okay. I'm going to pass the opening of the broth tube over the candle flame to sterilize it. And then I'm going to reaffix the lid. And I'm going to do this for S-Seri pH 5. So I will sterilize it by passing it through the flame. And then I'm going to add 1 drop. One drop to it. Pass the lid through the flame again to sterilize it. And then I am going to recap it. Okay. Remove the lids. It says to remove the lid of S-Epi pH 9 tube and pass it through the opening over the candle flame to sterilize it. So I'm going, this is number pH 9. Sterilizing this one. And then it says to add exactly 1 drop of the NaOH to the pH 9 tube to adjust the pH. Okay, there's 1 drop. I'm going to pass the broth tube to sterilize it 1 more time so I can close it. Okay. Then I'm going to do it to the S-Seri pH 9. Opening it and then sterilizing it. Adding 1 drop. Sorry. Okay, there's 1 drop. sterilizing it one more time so I can close it. So it says that the pH 7 broths are already adjusted to the pH 7 and require no additional additions of acid. 1 second, son. It says to wrap the HCI and NaOH pipettes in paper towels and place it in the trash can. So there we are. We'll place this in the trash can. And then it says, remove a graduated pipette from the cup of alcohol and shake it. So here's my pipettes. I'm going to shake it off. It says that using aseptic techniques, I'm going to pipette 2 drops of active S. epidermidis culture broth into each of the S. epi broths, the 3. So my S. epi ones are here. And then my culture broth is right here. So I'm going to do 2 drops of each of these. First I need to sterilize this. Make sure my pipette's dry. I'm going to okay, what we got there? My first one. I will sterilize this. Dropping 2 drops. 1, 2. Sterilize it so I can recap it. Now I'm going to do the same to this one. Sterilizing it. Doing 2 drops. 1, 2. Sterilizing it again so that I can recap. And then I'm going to do my last one, sterilizing it. Two drops. Sterilizing it to recap. I'm going to now place this pipette into a container of bleach. Now I'm going to do the same thing I did, but with the Cervice. Oh, let me put that back. Let me put this one, sterilize it so I can close this one. My active culture. Okay. Now I'm going to, sorry, my goggles are fogging up. Now I'm going to do it with my other one. Shaking off to make it dry. I'm going to get my other one, and I'm going to sterilize this one. Make sure I'm not touching the sides. I'm gonna go ahead and, oops, cap this one off. I squeezed a little too hard. And I'm gonna add 2 drops to each of my broths. First I gotta sterilize them in order to add the drops. Yeah. 1, 2. Sterilize it to recap it. Do the same for the next 2. My flame's running low, so I hope I can get this. 1, 2. Sterilize this one so that I can recap it. And my next one. Sterilizing this one. 2 drops. 1, 2. Sterilizing it. To recap. I'm going to place this pipette into the bleach container as well. And then I'm going to take a photo of my broths. And then return everything. And I'm going to let this incubate for 24 hours and then observe them and write down my observations. Thank you.